Molecular structure and basic properties of lysophosphatidylcholine and lysophosphatidylethanolamine. LPC and LPE are lysophospholipids derived from the partial hydrolysis of phosphatidylcholine and phosphatidylethanolamine. They are important intermediates in the lipid metabolism and maintain the amphipathic structure or dual nature properties of typical membrane lipids, a polar head and a nonpolar fatty acid tail. As for their structures, both LPC and LPE are built from a glycerol backbone, one fatty acid chain, and a phosphate group linked to a polar head group. This could be ethanolamine and choline. The fatty acid tail is a single hydrocarbon chain attached to glycerol via an ester bond. The phosphate plus head group, either choline or ethanolamine, are polar regions that interacts with water. Their functional groups include a phosphate group, which provides polarity and forms the hydrophilic head, an ester linkage which connects the fatty acids to glycerol, and an amine group found in the ethanolamine or choline head group. LPC is derived from phosphatidylcholine found in soy lecithin, egg yolk, and cell membranes. Meanwhile, LPE is produced from phosphatidylethanolamine, which is found in soybeans, animal tissues, and bacterial membranes. As for their synthetic sources, both can be produced or modified industrially for use in liposomal drug delivery and food emulsifiers. Both can be prepared by partial enzymatic hydrolysis or removal of one fatty acid from PC or PE using phospholipase A2. As for their biological role, they serve as intermediates in phospholipid metabolism. They also contribute to membrane remodeling and signaling pathways. They also act as precursors for resynthesis of diacylphospholipids. In some cases, they regulate inflammation and cell communication. Although they are smaller than their diacyl counterparts, PC and PE, LPC and LPE retain their amphipathic nature, allowing them to interact with lipid bilayer.
Unlike the stable two-tailed cylindrical shape of their parent phospholipids, LPC and LPE are single-tailed lipids, typically generated by phospholipase A2. These single-tailed structures give them an inherent cone shape, and their geometry is the key to their function. When this cone shape lysolipids transiently incorporate into one liplet of bilayer, they act as molecular wedges. This disparity in shape between the liplets creates significant positive spontaneous curvature stress. This induced curvature is not merely a structural side effect. It is a mechanistic requirement for all dynamic membrane processes involving shape change. Specifically, first, the vesicle budding and fission. The positive curvature induced by LPC and LPE helps membranes bend and pinch up to form transport vesicles. And signaling. LPC is generally a more potent curvature inducer than LPE due to its larger head group. These lipids are rapidly produced during cellular events like apoptosis or inflammation, making them not only structural modifiers but also essential bioactive lipid mediators that dictate cell fate.
Lysophosphatidylcholine or LPC and phosphatidylethanolamine are dynamic lipids that play vital roles in cellular energy metabolism. Lysophosphatidylcholine is a derivative of phosphatidylcholine that helps regulate lipids, transport, and mitochondrial function. It facilitates fatty acid delivery to energy-demanding tissues, and influence mitochondrial respiration by modulating membrane fluidity and signaling pathways. However, excessive LPC can disrupt membranes and impair ATP production, showing that its balance is critical for efficiency and energy metabolism. Phosphatidylethanolamine is abundant in the inner mitochondrial membrane that supports the structure and activity of ATP synthase and the electron transport chain. It directly contributes to ATP generation by maintaining membrane curvature and essential for oxidative phosphorylation. Together, LPC and PE maintain the flow and efficiency of cellular energy, one that directs lipid signaling and fuel supply, and the other that stabilizes the very machinery that produces ATP.
Lysophosphatidylcholine or LPC is not just a structural piece. It is also a key player in cell communication. It can bind to specific G protein coupled receptors or GPCRs like GPR119 and GPR4. This binding can trigger intracellular signals such as calcium mobilization, which influences processes like insulin secretion, immune responses. LPC also acts as a find me signal released by dying cells to attract phagocytes for cleanup. On the other hand, Lysophosphatidylethanolamine or LPE plays a significant role in delaying senescence in plants. It does this by inhibiting the enzyme phospholipase D or PLD, which is involved in membrane degradation during aging. In animal cells, LPE is involved in calcium signaling and can affect differentiation and migration.
Now, moving on to their protective roles, LPC helps maintain membrane integrity and acts as a signal for repair when damage occurs. It also supports surfactant function in the lungs to prevent alveolar collapse and aids in membrane remodeling in the liver during detoxification. Meanwhile, LPE improves membrane flexibility and assists in vesicle movement, helping protect the cell during transport processes. In plants, LPE helps membrane stay stable during stress conditions like heat or drought. In summary, LPC focuses on protection and repair, while LPE supports flexibility and stress resistance, working together to keep the membrane stable.
Everyone, today I'll be talking about the applications of Lysophosphatidylcholine and Lysophosphatidylethanolamine in medicine, food, and material science. In medicine, LPC is used in liposomal drug delivery systems like Doxil, which carries the cancer drug doxorubicin. With these liposomes help deliver the medicine directly to cancer cells with less side effects. Meanwhile, PE is very important in lipid nanoparticles or LNPs. These are used in mRNA vaccines such as the Pfizer-BioNTech COVID-19 vaccine. The PE derivative called Keto helps protect and deliver the mRNA safely into our cells. In food industry, however, both LPC and PE are known as lecithins, which act as natural emulsifier in products like chocolates and margarine, helping mixing ingredients smoothly. In material science, on the other hand, these lipids are used to make biodegradable films and biocompatible coatings, which are safe for both the body and the environment. That's all. Thank you for listening.
So this model presents several ethical, environmental, and economic considerations. So ethically, their use in biomedical research and therapeutic trials requires caution, as both are often derived from animal or human tissues, raising concern about sourcing, consent, form, and user safety. Environmentally, extracting these lipids from animals or marine sources may contribute to over-harvesting that may lead to resources depletion, while their chemical synthesis may involve solvents and byproducts that pose pollution risk if not properly managed. So economically, LPC and LPE hold significant value in the pharmaceutical and nutraceutical industries due to their application in drug delivery, metabolism studies, and lipid-based therapies. However, their production and purification costs can limit accessibility, creating a balance between innovation, sustainability, and affordability in their large-scale